Hi everyone. So today I'm sharing some of the, I would like to share some of my research regarding the preparation and characterization of this nanomaterial for cancer therapy. So first, in the domain of theranostics, there is an interest in developing drugs that have the ability to work as agents both for diagnosis and for treatment. And these drugs work in such a way that they have a targeting ligand attached that has specific affinity for a receptor expressed in cancer tissue. And once first the expression of this receptor is validated, that same drug can be used to treat the tumor. And while in the preparation of radiopharmaceuticals, there can be a lot of different isotopes that can be used for this purpose. Different isotopes, depending on their atomic weight, can have different different type of radiation emission, different degree of energy or tissue penetration that can be implemented for both imaging or treatment modalities. And while due to recent success in clinical trials, this has sparked a lot of interest in the development of new radiopharmaceuticals. And well, this leads us to the question of what can nanomedicines offer to this field, to this domain of these radiopharmaceuticals. And well, in research and recent publications, there's a lot of interest in nanomedicines for this field, specifically because they're able to offer multifunctionality by being able to chelate more than one radioisotope and offer both at the same time diagnostic and treatment in the same agent, and as well as having the ability to implement combination therapies by entrapping a chemotherapeutic drug, as well as having favorable tumor retention properties when applied intratumorally. And well, this leads us to the question, how should we design a nanoparticle that would be implemented as a radiopharmaceutical? And well, as you know, there are different ways that these nanomaterials can be prepared. And well, some nanomaterials, and usually what we see in some of these nanoformulations is a certain drug, a certain agent is prepared with multi-step synthesis with many purification steps. And many times these isotopes that are applied to a nanomaterial can be found on the surface or entrapped inside the nanoparticle. And while there are different ways that these nanomaterials can be prepared, if we are able to prepare one building block that is able to, the building block itself have the ability to work as a drug, as an imaging agent, and carry the radiotherapeutic agent, and this drug can be self-assembled into a nanoparticle. This would offer a more simple way of a simple model to prepare a more efficient radiopharmaceutical for as a nanomaterial. And well, this leads us to this technology that I've been working on recently, which is the texaphyrin macrocycle, the texaphyrin nanoparticle. So texaphyrin refers to this microcycle that you can see on the left side. It is a chelating ring. It has a 5 coordination pocket and is able to form stable complexes with heavy elements that are of interest in radiation therapy, such as yttrium, lutetium, bismuth, terbium. And well, this texaphyrin macrocycle can be prepared into a lipid that is able to form this liposome-like nanoparticles that could offer both abilities to target a specific receptor, as well as offering different modalities as photodynamic therapy, photothermal therapy, and has previously shown as being an efficient SPECT imaging agent or MRI contrast agent. And well, now we're trying to expand the reach of this technology to offer also radiation therapy. And well, this is how this nanoparticle has been prepared in the laboratory, where I'm synthesizing this texaphyrin lipid that contains this texaphyrin macrocycle as part of the molecule, and this is self-assembled by a microfluidic chip, by microfluidic mixing to form this nanoparticle. And while the aim of my project is to characterize this yttrium nanotexaphyrin that can be targeted specifically to a tumor model and validate its ability to work as a radiotherapeutic agent. And while the first part of my study was to validate its chelation, which is what I worked on first after the synthesis, and I was able to efficiently show that this microcycle was able to chelate yttrium in conditions that are similar to that which we see in a radiolabeling laboratory. And once this chelation was validated, this nanoparticle was prepared by was prepared, was self-assembled. These texaphyrin building blocks were self-assembled, and I would characterize it first by absorption spectra and fluorescence spectra, showing it has different spectrums based on the when it's intact as well as when it's disrupted, and as well we can see that it fluoresces, and once the nanoparticle is disrupted, we can characterize its fluorescence emission as a disrupted nanomaterial. And this disruption to fluorescence can be used to characterize its stability. So we can see that it is stable over a period of 64 hours in conditions that would mimic conditions that we would see in vivo, and as well as the chelation stability, which is one of the most important tests that I did in order to see that this yttrium metal that is chelated inside the texaphyrin stays chelated, which is extremely important once this drug would be applied to an animal model. And while after this, I also characterize this nanomaterial using both seismic morphology, using both transmission electron microscopy and dynamic-like scattering to show an average size of about 89, 90 nanometers with a PDI lower than 0.2, which shows some homogeneity in the sample. And while this nanoparticle formulation contains as well this targeting ligand to prostate cancer cells that I'm currently in the process of validating, and I have some new data that I'm showing here regarding this time-dependent uptake to these prostate cancer cells that is promising to show the evidence that this nanoparticle is being uptaken by these cancer cells before applying the model this to an animal model. And well, just as my conclusions for what I'm presenting here is that I've been able to prepare this nanomaterial with conditions similar to the way we see as a radiolabeling conditions, in advance to working with the radioisotope itself, which is actually what I'm currently working on. And after the preparation of this radioactive nanoparticle, my plan is to first prepare this to show the biodistribution of this drug in an animal model and get the first therapeutic data. And that's what I would like to present. And I would like to thank all the people in my lab and the agencies that have helped me work on this research. Thank you.
Thank you, Alberto. I'm pleased to open the floor to questions from the audience.
Thanks, Alberto, for a really fantastic presentation. Really like the research that you're doing. I have a question. You said that you checked the chelation stability. My question is, how did you check that? And did you also check the metabolites, or are you planning on checking the metabolites once you proceed to animal studies? Thanks.
Thanks for your question. So yeah, the chelation stability, I did it using liquid chromatography and mass spectrometry. So what I would do is that I would prepare the nanoparticle itself and get a reading of I would characterize the spectrum of the nanoparticle immediately after the chelation. So I would see once it's 100% chelated, how much of the chelated texaphyrin I can detect using mass spectrometry. And later on, I would place this formulation of the nanomaterial in a solution of fetal bovine serum, and I would leave it for either 12 hours, 24 hours, 64 hours. And at each of these time points, I would check the mass spectrometry again of the sample, and I would detect if I can find any unchelated texaphyrin. So if I see any increase in the amount of unchelated texaphyrin to the time point equals 0, then I would get that difference, and that would be how much of the texaphyrin became unchelated over that period of time. So I did it by mass spectrometry. And your second question was about metabolites. Is that
yeah,
for in vivo studies?
I guess, yeah. I'm asking whether you're planning to do that, or do you think it will be different once it's in vivo, or not? Like the chelation stability, would it be different in the in vivo systems than it was in the serum?
Right. So I would, the characterization of the chelation stability, I would do it in the serum, mimicking conditions that are done in vivo. For the first in vivo studies, what I'm planning to do is getting the first degree of therapeutic efficacy using biomarkers that are markers of cell death from double-stranded breaks. So that's what I would first try to do with the in vivo studies. And for the chelation stability, I would characterize that using the studies I showed in this presentation.
Okay. Okay. Thanks, Alberto.
Thank you, Nashmia. Marshall, any other questions from the audience?
Presently, no other questions.
All right, I have one for you, Alberto. So what was your reasoning to choose yttrium as the metal to prepare the nanomaterial?
Right, so we're first trying to prepare this proof of concept study to get the first case of therapeutic data using this nanomaterial. And we're choosing yttrium because it's, well, it's a metal that's been used previously in effectively in clinical trials. And it's something that it's a metal that I've been able to show that I'm able to chelate using the same conditions I would get in a radio labeling setting. And to get the first examples of therapeutic efficacy, it's the most optimal metal to use for a proof of concept study.
Thank you so much.